So he graduated with the Bachelor of Science in Kansas State University. You have a lot of degrees, by the way.
I've been at a lot of different universities. I've been thrown out of some of the finest institutions.
And he got his Master of Science in Entomology in University of Arizona, and he got his PhD also in entomology in University of Florida. Then he did a series of postdocs in Mississippi State, also in UC Davis.
I was here as a postdoc.
Who did you work with?
I worked with, with Bob Washino and with Bruce Eldridge.
Yeah. And then he moved to the NIH as a MacArthur Fellow, and he's been working on malaria research since then. So he also moved from institutions as a professor. He was at University of Texas, and then he came back to University of California, Davis, and where he's been for the last number of years. He was in our department for a while, and then now he's in the vet school. He was the director of the Center for Vector-Borne Diseases a while ago, and then he retired from that position. So if you know Greg, he teaches with me, so I hear stories from him all the time. He has tons and tons of crazy stories about going to countries, doing research where he needs military escorts. He has stories about fighting monitor lizards, like 4-feet lizards with mattresses and crazy things. But anyway, I'll let you tell him.
Yeah, this is kind of a crazy story, so it fits in okay. Can everybody see this? Should I turn the lights down a bit? So thanks, Joanna, and thanks to the entomology department for a chance to come back and talk a little bit about some of the things that we've been doing lately. So just as some background, and I'm sure this audience is familiar with malaria and the fact that malaria is a very serious problem. It's one of the most important infectious diseases of man globally. It's certainly the most important insect-borne human pathogen in terms of its impact. Roughly 50% of the world's population is at risk, live in places where malaria transmission is going on. It's estimated that there are 225 million clinical cases of malaria every year, and that's probably a gross underestimate. And there are roughly 800,000 deaths per year due to malaria. That's primarily in children under the age of 5 and pregnant women, and most of this is in sub-Saharan Africa. So just a quick background of sort of the state of where we are at with respect to controlling malaria with approaches that are aimed at managing the vector species, which is really the best public health management tool that's available. And so you can see that what's currently being done all across sub-Saharan Africa are two approaches. One is spraying inside people's houses with residual insecticide, and that's primarily DDT. And then the use of insecticide impregnated bed nets that people sleep under. And these nets are impregnated with pyrethroid insecticide. So you can see that really with respect to the technology that's being applied for managing insects, it's nothing new. These approaches have been around for a very, very long time. There are some new approaches that are being developed, a number of different things, some new chemistries for insecticides. There are a number of different genetic-based control strategies that are being worked on. But as of right now, this is the main thing for controlling malaria in sub-Saharan Africa. Of course, the problem is with insecticide resistance, and resistance to both the DDT and pyrethroids, and that's mainly because it's cross-resistance. I'll talk a little bit more about that later. But you can see from this map that was put out in the malaria report from the World Health Organization is that essentially every country in sub-Saharan Africa is reporting resistance in malaria vectors to both to pyrethroids and DDT. So this is a big problem. Okay, this is the mosquito we work on, Anopheles gambiae. It is the principal vector of malaria in sub-Saharan Africa. It's probably the most effective malaria vector in the world. It's very susceptible to infection with Plasmodium falciparum and Plasmodium vivax. It is a peridomestic mosquito, so it's associated with people. And one of the things that contributes most to its sort of super effectiveness as a vector is that this species feeds almost exclusively on man. So even when there are large populations of large mammals around, cattle, sheep, this mosquito, if you take a sample and do a blood meal analysis, you'll find that over 95% are feeding on people. So it has a very strong affinity for biting man. This is basically the distribution of malaria in sub-Saharan Africa, and I put this up just to show that malaria transmission is active across a very wide range of ecological settings. The only places really where there is little or no transmission is at very high elevations. Other than that, malaria transmission is going on in very dry places, so places that are adjacent to the Sahara Desert, for example, all the way to rainforests in southern Cameroon, for example, in the Congo. These are the places where we have worked recently over the last several years, and I'm going to focus on sites in West Africa. So Guinea-Bissau, which is here, Mali, which is where we've been working for the longest amount of time, and Mali is, let's see, from about here north is the Sahara Desert, so there's very few people, there's hardly any water, and consequently there are very few mosquitoes and very little malaria transmission. But the southern half of the country is relatively wet and is hyper endemic for malaria. There's a lot of malaria in southern Mali. Cameroon also, although there's transmission occurring across the entire country, it goes from very dry habitat adjacent to Lake Chad in northern Cameroon to rainforest in the south. Okay, so what we work on is looking at the population genetics of Anopheles gambiae, and those of you who are even a little bit familiar with the species know that there are two subpopulations of this mosquito that exist in West Africa. They occur in sympatry in many places. They're morphologically identical. There is a PCR assay that can be used to distinguish them. So S form shows a band here. This is based on polymorphism in the ribosomal DNA locus, which is on the X chromosome. This mosquito has 3 pairs of chromosomes. It has an XY sex determination system. So there's females are XX and males are XY. That M form, it's a fainter band, but it has a very distinct, migrates to a very distinct position on a gel. And hybrids can be recognized, at least hybrid females. Obviously, since this is an X-linked gene, you wouldn't be able to detect a hybrid male. And I should say that these are hybrids that were generated in the laboratory. Where these things occur in sympatry, there's very strong assortative mating. And that's really going to be the subject of what I'm going to talk about. Recently, just this year, the M form was elevated to species status, Anopheles kalutziae. So these two are now recognized formally as distinct species. As I mentioned, they occur in sympatry over most of their range. Okay, but there are some ecological differences that characterize these things. So The M form is associated with water that's more permanent or semi-permanent, and it's associated with water used in agriculture. So you find it in irrigated agriculture where water is standing, and the main thing is rice. The S form is more, the larvae breed more in sites that are temporary, rain dependent. So puddles in roads like this, or in fields that get flooded after a rain. So their larval habitats are different. If we look at the distribution broadly in Mali on the left and in Cameroon on the right, you can see that they're not distributed randomly. So the M form predominates in northern Mali, where it's drier. The S form predominates in southern Mali, where it's wetter. In places where there are distinct wet and rainy seasons, the relative abundance of M and S changes, with the M form predominating during the dry season and the S form predominating during the wet season. Likewise, in Cameroon, the spatial distribution is they're not randomly distributed in space. And what you see is on these pie charts in yellow is hybrids, and you basically don't see any. So even where these things occur in sympatry, hybrids have been rarely identified. Both in Mali, the frequency of hybridization has been estimated at less than 1%. And in Cameroon, it's felt that there's no hybridization at all between the two forms. Another very interesting thing, and to get back to insecticide resistance, we did a study some years ago looking at several populations in southern Mali, indicated on this map, where M and S forms are sympatric. And we were interested in looking at the distribution of a KDR gene, which is a gene that imparts resistance to pyrethroids and DDT. It's a mutation in the sodium channel that these insecticides bind to, and that basically is how they work, their mode of action. Interestingly, we looked at these four populations. So we have the distribution of genotypes in the M form on the left side in each of these graphs, and in the S form on the right side. And what you can see is that the M form that populations that were assayed, there was the frequency of KDR was 0. There was no KDR gene at all in those populations. Whereas in some places, for example, here in Pemperena, almost every individual carried at least one copy of the KDR. So there's a big difference in the distribution of this insecticide resistance gene between these forms as well. Okay, so what we were interested in is looking at taking a closer look at how much gene flow there is going on between these two forms. Because the problem with using a single gene marker to distinguish forms like it, it's dicey. Let's just put it that way. For one thing. And secondly, if you're interested in looking at hybridization and introgression, having only a single locus is problematic, because if there's backcrossing going on, you're going to be missing a significant proportion of hybrid due to that. So this is what is currently understood with respect to how much hybridization is going on. As I mentioned, in Cameroon, it's not thought that there's any gene flow between M and S. In Mali, it's about 1%. But there are sites in coastal West Africa where hybridization occurs at frequencies as high as 25%. So if you went in and sampled the population, you'd find 25% of those individuals are hybrid. And so there's a spatial heterogeneity with respect to how much hybridization is going on. It's thought that the reproductive isolation is primarily has to do with mate choice, so it's mating reproductive isolation. You can produce hybrids in the laboratory, and they are fine. They're sexually fertile. The sex ratios are normal. Their longevity is normal. Their fecundity is normal. You can backcross F1s in either direction, and backcross individuals likewise show no sign of any hybrid fitness, any decrease in hybrid fitness. But it's also generally thought that in nature that may not be true, and frequently, of course, it's not, because you're not replicating the natural environment in the lab. Okay, so the kind of, from an evolutionary perspective, what we're interested in is sort of looking at issues surrounding the idea of species evolution. And typically, sort of the classical definition of what a species is populations that are reproductively isolated, and it's best if they're completely reproductively isolated. That makes them good species. But we know, in fact, that the boundaries between species is generally or very frequently porous. So there is hybridization going on. And we know that even a small amount of gene flow will have the effect of homogenizing populations. So the question, or one question in evolutionary genetics now that people are interested in, is how do you evolve and maintain genetic divergence in the face of gene flow? How does that work? How can that work? Okay, and the answer is, or at least one, is that divergence can happen at some genes, even if there's gene exchange going on at other genes. So in other words, you can have some genes moving between populations and other genes not. So some genes, movement of those genes is opposed by selection against heterozygotes, basically. So that this has been formalized by a number of different people in a number of different ways, but this is probably, at least from my perspective, the best idea. And this is what's called a genic model of species differentiation. So if these are two genomes, if these bars represent two genomes, and where they're this green color, they're identical, there may arise certain number of loci where there's no gene flow, where those genes are not moving between these populations, despite the fact that we may get movement in other regions of the genome. And it's thought that these loci contain what are called speciation genes, and that is genes that are basically affecting reproductive isolation. It may be in the form of mating behavior for pre-mating isolation, or it may be that the heterozygotes have a decreased or are not as adaptive to the specific habitat that these are living in. And it's generally thought that these genomes are coming from populations that are residing in slightly different habitats. So one is exposed to a habitat and is developing adaptations specific to that habitat, and the other some different habitat and a different set of adaptive genes. These regions will expand or may expand as mutations arise near them that have some sort of epistatic interaction that improve the phenotype. And they will continue to expand by hitchhiking, for example, until eventually you get two widely differentiated genomes. So speciation occurs sort of in a continuum. Okay. Now, the other, well, I think I have an illustration. Yeah. You would expect that these islands of divergence, or speciation islands, would arise in regions of the genome where recombination is suppressed, so that multiple genes are not being upset by recombination. So you would expect to find them, for example, around the breakpoints of chromosome inversions, where it's known that recombination is suppressed. and proximal to the centromeres, where recombination likewise is suppressed. So that would be where you would expect to find these things if you were looking for them. Okay, so in fact, a number of studies now have been done comparing the genomes of the M and S forms in a number of ways. The first studies actually were done here at Davis in Sergei Nuzin's lab by Tom Turner, who was a student of Sergei's at the time. And then a number of studies have come on afterward that support this idea. And he describes genomic islands of speciation in Anopheles gambiae, in the Anopheles genome. So if you compare M and S, and he used a microarray to do this, it was pretty primitive by today's standards, but it had roughly 150,000 markers on it that across the whole genome, or across most of it. And what he found was that that divergence between M and S is concentrated in 3 small regions, all of them very close to the centromere. And he described these as islands of speciation. So he said, well, I'm going to come to that. So they're saying that and the M and S forms on this continuum would be somewhere around here. Okay, in a relatively early stage of speciation, if you want to think of it that way. There was some work done subsequently that basically produced data that refuted this idea that there are islands of speciation, or that such things are important with respect to this M and S form. And this sort of model has been called the incidental islands of divergence model. This was done by a group at Notre Dame, mainly by Brad White, who's down in Riverside and was here not long ago to give a seminar. So basically what he did is he identified a single marker in each island. Okay, so 3 independent genetic markers, SNPs, in each of the 3 islands, so that he would be able to recognize parental genomes, F1 hybrids, which would be heterozygous at all 3, and then backcross individuals, which would be sort of a mosaic. And he assayed population. He looked at a total of 517 mosquitoes collected from various sites where M and S are sympatric. And what he found was overwhelmingly they were either pure M, they had M SNPs at each island, were homozygous for M SNPs at each island, or S. He found 3 that had genotypes suggesting that these were F1 hybrids, and only 2 that looked like they could be backrows. And so what he's saying, what he proposes is that hybrids observed in nature are mostly F1 hybrids, and that F1 genotypes are effectively sterile. So basically, you may have some level of hybridization, but it's a dead end. Those F1 hybrids are not going on to backcross, and so you're really not getting introgression. So these guys are saying that in fact the M and S forms are more here. There was also some work done where they compared genomes by sequencing, and they found their interpretation was that divergence was wide, was significant, and distributed over the entire genome. There were some problems with that study. For one thing, they used laboratory colonies rather than mosquitoes from nature, so that's probably not a great idea. Okay, so to summarize, we've got 2 models explaining the evolution of these M and S forms. The speciation island hypothesis, which says that differentiated regions contain loci that are responsible for assortative mating or ecological differentiation. So these are then speciation genes. And that the rest of the genome is neutral with respect to species differences, or at least close enough to neutral so that gene flow that is going on is keeping the remainder of of the genome homogeneous or undifferentiated. Okay, if you look at the total amount of the genome that's contained within these islands of divergence, it's about 3%. So 97% of the genomes of these two species are essentially identical. The incidental islands model says there's actually very little gene flow between M and S forms, that the low level of differentiation on the chromosome arms is due to segregating ancestral variation, not contemporary gene flow, that hybrids in nature are mostly F1 and these die before they reproduce, and therefore F1 genotypes are effectively sterile, and that realized gene flow between the forms is nearly zero. There's some limitations with this work though. All the comparisons prior to the one that White did with those 3 SNPs were using just a single X-linked locus, and that's problematic. Assessments of hybridization frequencies and comprehensive tests for introgression are precluded from those studies, from the majority of those studies, because they use DNA pools rather than individuals. The sample sizes were small, or because M&S laboratory colonies were used. So we thought it was worth taking a closer look at this. And there were several questions we were interested in answering. Just how much hybridization is occurring in natural populations? What is the spatial distribution of hybridizing populations? Is it restricted only to coastal West Africa? Are high levels of hybridization restricted only to coastal West Africa, as has been observed? We're interested in determining whether hybridization in fact does mean gene flow. In other words, is there mating going on beyond the F1 generation? And we were interested in looking at hybrid fitness in nature. So what we did, we basically extended the approach that White took. So we developed a panel of SNPs that we call divergence island SNPs. A total of 14, 7 on within the X chromosome island of divergence, 4 on the left on chromosome 2 in that island, and 3 in the island on chromosome 3. Okay, so and these are fixed differences between M and S. So we were able to generate multi-locus genotypes that would allow us to define the forms. So an individual that's homozygous for all 14 M-form SNPs is a pure M individual. And likewise, if it's homozygous at all 14 loci for the S-form SNPs, it's a pure S form. F1 hybrids are heterozygous at all 14 loci. And then backcross individuals are a mosaic. So we analyzed populations at these sites across West and Central Africa. The dark brown color is the abundance of the M form. The tan color is S form, and the 2 green colors, light green are F1 hybrids and dark green are backcross. And basically what we found was that, in fact, hybridization is occurring at a much higher rate than previously thought. Every population that we looked at where M and S occurred together had a frequency of hybrid individuals that ranged between 5 and 97%. Okay, so this notion that rates above 1% occur only in coastal West Africa are not true. So we found that patterns of hybridization did vary spatially. The amounts of hybridization were different in different populations from different places. We're assuming that this has to do with differences in the environment, so that environmental cues that are involved in mate choice are more effective in supporting assortative mating in some And so we assume that if this is true, that levels of hybridization at a single site that varies environmentally over time should show fluctuations in the rate of hybridization. And so in the following series of slides, I'm going to show you data that we collected from a single site in southern Mali, samples taken over a 20-year period from 1991 to 2011, looking at specifically at patterns and frequencies of hybridization, evidence of introgression, and evidence to support the idea that hybrids have reduced fitness in nature. The way that we did this is we arranged each individual mosquito genotype in a row. We used different colors, so dark blue to represent S, light blue to represent M, and yellow are heterozygotes at each locus. We compressed those down into bars for each individual, sort of looking like this. And then for our population, those bars were just stacked. So you would be able to visualize, look at a population, and see the distribution of genotypes relatively easily. And these are the 14 loci. Okay, so we starting in 1991, we found a pattern that suggested both in 1991 and in 96 that the boundary between M and S is very strong. The relative abundance of M and S is different between years, but that was known. It was known that the relative abundance of forms changes from year to year at most sites where they're sympatric, so that was really nothing new. We did find evidence of past hybridization. And I should say that, just to reiterate, I don't have the data here, but we did analyze allopatric populations of M and S. And so the polymorphism, these are indeed fixed differences. So these are not ancestral polymorphisms. So it looked like to us that we had very strong barriers, no evidence for ongoing hybridization at the time these were collected, but evidence that there had been some hybridization in the past. In 2002, the pattern was very different. In fact, this time we did find hybridization. It occurred at a frequency of about 0.8%. Okay, but along with that, we found a much higher abundance, around 11%, were backcross genotypes. So we're seeing a pattern where in some years there's no hybridization, and in others we're getting hybridization at least at around 1%. Now in 2004, 2 years later, our sample, most of the backcross individuals are gone. So the frequency of backcross individuals dropped from 11% to 5%, suggesting that in fact those genotypes were selected out. And that, in fact, hybrid genotypes do have reduced fitness. But then, really interesting, in 2006, these mosquitoes were having a party or something. I don't know what was going on, but the F1 hybrid frequency was 12%. So assortative mating was breaking down big time in 2006. What happened subsequently is the thing that is really very interesting. So after this round of a very high level of hybridization, where the reproductive, the pre-mating barriers appear to have broken down to a large extent, for the X island and the island on chromosome 3, the hybrid genotypes appear to have been selected out. Strong assortative mating seems to have been reestablished because we find no F1 hybrids in these samples. But the island 2 seems to have introgressed completely from the S form into the M form. And if you look at the frequencies of the SNP genotypes on chromosome 2, those 7 SNP genes, they're in equilibrium. So it appears to be a stable introgression of this island 2. So we conclude that hybridization well in excess of 1% is occurring throughout the sympatric range of M&S. Hybridization does in fact result in gene flow between the forms. The rates of hybridization fluctuate temporally. Hybrids do have reduced fitness, but not to the level that would exclude introgression. And the islands of speciation model, we think, based on these results, is the best hypothesis to explain the relationship between the M&S forms. Now, obviously, what happened here is a question. Why was there a breakdown of assortative mating? And we collected what data we could, what environmental data that was available, and nothing really jumped out. It wasn't exceptionally rainy or exceptionally dry. There were no human projects undertaken in the area that weren't already there and had been for quite a long time. And then, why do we get this introgression of the island 2? And that one we think we do understand. If you look at, this is data from the World Health Organization on the distribution of insecticide impregnated bed nets. So prior to 2005, there were impregnated bed nets were not being used in Mali. Starting in 2005 and really increasing in 2006, bed nets were being distributed big time across the country. And this, for us, coordinates right with the time that we're starting to see this pattern of very high levels of introgression, especially on that island 2. So between 2006 and 2012, nearly 5 million insecticide-treated nets were distributed across Mali. Okay, this is where we have our sample where we're starting to get hybridization. And you can see, as I mentioned earlier in that paper by Fred Trappe, the S form here in blue had a relatively high frequency of KDR at the time that they were sampled in 2004 for that study. It was 0 in the M form. But after 2006, when bednet use is increasing, and when we're getting introgression of this island 2 from the S form into the M form, we see the frequency of KDR shooting up. In fact, the KDR locus lies within that island 2. If we look at the allele frequencies of KDR in the M and S forms from the samples that were used to generate these data, we see that in 2004, the M form had a frequency of 0. It was about 60% in the S form. At the time when they were hybridizing, just taking pure M and pure S, the frequency of KDR in the M form is starting to increase a little bit, a half 5%. 50% in the S form. And then in 2010, we find the frequency of KDR jumping to 80%. So at a frequency of 0 prior to this introgression, and at a frequency of 80% 4 years after that round of introgression. Okay, we also determined the genotype frequencies. So the M form were homozygous susceptible. The S form had frequencies around 33% susceptible, 41% heterozygous, and 26% homozygous KDR. We looked at the genotypes in just the F1 individuals. So we only took those individuals that were heterozygous for all of our divergence island SNPs. There were 17 of them. We looked at the observed and expected frequencies, and there was no significant difference between them. So with respect to KDR, whether or not an individual had KDR or not didn't seem to be influencing mating at all. Now we looked at just back-crossed individuals. So these are basically individuals that are M form on the X and 3L, but either S form or heterozygous for this 2L item. And these were the numbers of individuals that we sampled from each of those sets of individuals. We looked at the observed and expected frequencies, and what we found was that this is the frequency of wild type. So you can see these are the expected frequencies. Although these are not frequent, these are the expected numbers based on the sample size. So we expected to see 4 homozygous susceptible. We observed only 1. In 2011, we expected to see 2. We saw none. In the 2012 sample, we expected to see 6, and we saw none. So basically, in fact, the susceptible, those individuals that are homozygous susceptible for KDR are being selected out. Finally, we looked to see how much of the genome was being affected by this. And remember, the centromere is here, so the centromere would be here. So this blue color are S-form KDR homozygotes. The light blue are M-form wild-type individuals, and the others are back-crossed individuals. And so what you see is that it appears that the strong selection on KDR is affecting this entire island. So you see, what you see happening, let's see, in these M-form individuals that were introgressed, that they're very distinct. Or excuse me, these are pure M-form individuals, wild type, homozygous wild type. They're very distinct from S-form. This graph is showing proportion of S-ness. So you're more S as you go to the top and more M as you go to the bottom. But the differentiation over the rest of the genome is essentially nothing. So strong selection on KDR appears to be pulling this entire segment. It's roughly, let's see, it's about 3 megabase pair section of the genome along with it. And if we look for evidence of selection on this, or a selective sweep, we see it. So this is Tajima's D statistic. And you can see right at the KDR locus, variation is way down, suggesting a selective sweep acting on that gene. So to conclude, the KDR mutation does not appear to be related to the breakdown and assortative mating. So they appear, when we're getting hybridization between M and S, the KDR genotype doesn't seem to be affected. Insecticide resistance is a selective force driving introgression of a roughly 3 megabase pair region of the left arm of chromosome 2 from the S form into the M. And that, I should back up, and this is interesting. The introgression is asymmetric. So if I'm S form and I have the KDR gene, and I'm M form and I want to get that gene from you, then hybridization or introgression should be going in that direction, preferentially, and not the other way. So S form doesn't want susceptible genes from the M. And in fact, these results show that it is asymmetric. So it's introgression from S into M and not the other way around. We think that this, and we're excited about this because we think this, the information that I just showed you, is probably the best example of adaptive introgression in an animal species. So adaptive introgression is a theoretical thing that's been observed a lot in plants, but there are not, with the exception of maybe warfarin resistance in mice going across species. I don't think there's a better example than this, so we're pretty excited about this from that perspective. Okay, so my acknowledgments. Yusuk Lee is a faculty in PMI who works with me and Anton Cornell. She did the island genotyping study, and I'm happy to say that that should be out within the next few days in PNAS. Laura Norris is a postdoc in my lab from, she did her PhD at Johns Hopkins. She did all the KDR population genetics work, and a lot that I just didn't have the time to show you, estimating selection coefficients and doing some modeling and stuff. Brad Main is actually a relatively recent postdoc in my lab, being supported off the NIH training grant that we have. He did his PhD with Sergey Nuzden down at USC, and he's doing a genomics analysis to look at just how much of the genomes are actually introgressing, and it's very interesting. It's preliminary, but it looks like it's pretty much just that section of chromosome 2. What's also really interesting here is that if you look at the introgression and compare the X chromosome, chromosome 2 where the KDR is, and chromosome 3, it seems like the M form has picked up that chromosome 2 island with the KDR, but all the other introgression on X and chromosome 3, it reverts. Those individuals are selected out. So it's really it's really an interesting story. It's amazing that these things could do this. And a mechanism is something that we're trying to figure out. And then finally, Anton Cornell, who does cytogenetics for all of our work. And I didn't show data with respect to chromosome inversions, but chromosome inversions also have a pretty profound effect on the distribution of both polymorphism and divergence in genomes. That's nothing new, but it's something that needs to be accounted for if the species you're working on has a lot of chromosomal polymorphism, which this one does. I'd like to just acknowledge some other people in the lab. Allison Weekly, who's our lab manager, and Caitlin Neiman, who has done a lot of the routine bench work towards generating these results. Claire Marsden was a former postdoc. She's now down at UCLA. She did work in Guinea-Bissau, a lot of which I did not show. Michelle Slotman is now an associate professor at A&M. He was a postdoc in my lab, and he did work looking at recombination rates on the within that X island. Some really nice real genetics, actually. He did crosses and counted genotype frequency to estimate recombination rates. Fred Trappe, sitting there next to him holding a beer, is the guy that did the distribution of KDR in those different, in M&S forms from those 4 different villages in Mali. He's now a senior lecturer at Keele University in the UK. And of course, I have to acknowledge our collaborators in Africa. Wouldn't be able to do any of this without their help. We work with Mali Research and Training Center at the University of Bamako in Mali, with Abu Fofana and Sheikh Traore, who's the head of the entomology department there. National Institutes of Public Health, the malaria control program. In Guinea-Bissau, Amabilia Rodriguez is the director, and João Denise is the entomologist. And in Cameroon, the National Malaria Control Program, Etienne Fanjo, is an entomologist, but he's also the director of the National Malaria Control Program, which is, if you know anything about public health in the third world, he's not an MD, he's a PhD entomologist. It's pretty cool. And Solomon is the entomologist. And that's the end of my story. Thanks.
period of increased hybridization occurred also at a time when the relative frequency of the two forms was more equitable. There were larger, relative amounts of S.
Yeah, if you look back,
Frequency of the two forms next to
it doesn't.
it does not.
Yeah, we looked, because you would think that if one was really rare, it's kind of hard to find a mate, so you go with what's available. But there's no, we couldn't find any correlation between the relative abundance. No, but that's one of the first things we thought of too.
It looked like that was the case in 2006, that they were kind of more even.
They were pretty even.
Yeah.
Yeah, they were. But they had been in the past as well, and there was nothing. So, and then at a lot of the other sites, the same thing. I didn't have time to show those diagrams for all the other sites that we looked at. And there's also some really interesting things happening in other places. But just not enough time to do that. That work is described in this PNAS paper. That should be available like tomorrow, maybe. Yeah.
Well, again, I can't follow. The M&S thing intrigues me because you say you have these allopatric populations that are pure M&S.
Yes.
So I mean, it's what do I started thinking at the beginning of this talk? I mean, it's conceivable these things did just arise like good old-fashioned allopatric speciation a long time ago, right?
Yep.
And then now become broadly sympatric.
Yeah, in fact, that's my favorite idea.
Yeah.
But not,
It's the other like one.
not this one.
Yeah.
Oh, this. Simple. The new buildings we're in over at the vet school, everything is some kind of really fancy. So I won't around. I'm an old-fashioned guy. I like this. Yeah, I agree with you. In fact, there's a little sort of. I had a section at the tail end of this paper that's coming out soon, talking a lot about that, but they maybe take most of it out. I left. There's enough in there though that you can see that that's what I think. So I think it's a good point when we talk about a speciation continuum. I think it's important to realize that populations can move in both directions. So, and I think in this case, my sort of pet hypothesis is they moved pretty far, but now at various places in, for example, in coastal West Africa and at these sites, they're moving back. What do you think about considering these two things distinct species? I wasn't going to get pulled into that.
Yeah, I think it's reasonable given this interpretation of the past history. I mean, I think many people now are going back to sort of more or less reproductive data rather than reproductive data. The more or less.
Well, in medical entomology, that's you got to have reproductive isolation. Boom. But I spent too much time around Jim Mallow when he was visiting. So my attitude about that's changed a little bit. He's on the other extreme, I guess. Any other?
So those bed nets not effective anymore or not effective?
Bed nets are starting to be not really good. There's some other things happening as well, which I didn't talk about, and we are interested in fact working on. What people are observing is that the feeding time for Anopheles gambiae is changing. So typically they feed in the middle of the night. So the peak activity is like after midnight. And everybody in these villages is in bed. I mean, under a bed net, they've got a bed net. What's happening is we're observing that they're feeding earlier. They're starting to feed earlier, like around 7, 8, they're starting to feed before people are under the nets. So that's happening as well as this KDR gene. So they're getting both sort of classical insecticide resistance and some what appears to be behavioral resistance. So yeah, bed nets, and really, there's behind that, there's not much. Bed nets have saved many, many, many lives since they've been instituted, but I think most of the entomologists who were looking at this early on were saying this is going to be great for a while, but sooner or later these things are going to develop resistance, and you're going to have a problem. And sure enough, that's starting to happen. Okay, thanks.